functional anatomy of prokaryotic and eukaryotic cells. So we're going to compare prokaryotic and eukaryotic cells. I'm hoping that a lot of this is going to be review. Some of it might be new, but most of it should be review. Oh, there's somebody. From biology. So prokaryote comes from the Greek words for pre-nucleus, and eukaryote comes from the Greek words for true nucleus. Just kind of a fun fact, but basically one has nucleus, one does not. So here's an overview of the differences between prokaryotes and eukaryotic cells. Prokaryotes have a circular chromosome that's not bound into a membrane. When you look at the chromosome of a prokaryote, sometimes they draw it like pretty crazy all over the place, but if you were to pull it all apart, tease it all apart, it's a perfect circle. Prokaryotes do not have histones, so they're not going to package their genome by wrapping it around those protein histones. Prokaryotes do not have any organelles, nothing that is membrane bound. For prokaryotes, we have different types of cell walls. Bacteria always have peptidoglycan. Well, there are two exceptions to that rule that we'll see in a little bit. But generally speaking, bacteria will contain peptidoglycan in their cell wall. And then archaea will have a pseudomurin cell wall. And they divide by binary fission. So one cell becomes two. Now, eukaryotes have paired chromosomes, and these are in a nuclear membrane. So all the genome is stored into the nucleus. The chromosomes are wrapped around histone proteins. The DNA, is wrapped around histone proteins. This is to package more DNA. Eukaryotes are more complicated than prokaryotes, so they're going to have more DNA. Eukaryotes have organelles, and we're going to go through all of the different types of organelles. Again, I'm hoping that's a review. If it is not, let me know, because I need to know what you learned in biology. And then we have polysaccharide cell walls when they're present for eukaryotes. So not all eukaryotes have cell walls, but some do. So fungi cells and yeast have cell walls, and those are eukaryotes. So that's just something to keep in mind. A lot of people get that wrong when they're giving the differences between eukaryotes and prokaryotes. They say that eukaryotes never have cell walls. Well, that's not true. Animal cells don't have cell walls, but fungal cells, yeast cells, those have cell walls, and they're going to have chitin in their cell walls, which we'll look at in a little while. Eukaryotes also divide by mitosis, so they're going to have a different and much more complex division. So when we look at the size, shape, and arrangement of bacterial cells, the average size is between 0.2 to 2 micrometer in diameter, and that's by 8 micrometers in length. So we are talking about three-dimensional creatures, right? Most bacteria are monomorphic, meaning single shape, but some of them are pleomorphic, which means they have many different shapes. So this is one of those reasons why we often characterize, or we do our taxonomy for bacteria based on their ribosomal RNA rather than their shape. Now, they used to be characterized specifically by their shape, but because we do have some pleomorphic bacteria that can change their shape on you, we really rely on that ribosomal RNA to group bacteria. So let's take a look at some of the size, shape, and arrangement of the bacterial cells. We have bacillus. These are rod-shaped, and there's a picture of bacillus over there on the, what side is that? That's the right-hand side, sorry. Coccus are spherical shape. We're going to look at a picture of those in a little bit. Then we have a number of different spiral shapes. And the spiral shapes include Vibrio, Spirillum, and Spirochete. And the Vibrio is at the top there. They kind of look like half moons. Spirillum, just like their name implies, they're little spirals. And the spirochetes are really, really thin spirillums. And when they move, their movement that they have through the water almost looks like a worm, the spirochete. The spirillum definitely has a corkscrew twist to it when it moves. We can also have some different really fun shapes, star-shaped and rectangular. Now those are pretty rare, and generally you're going to find those in an aquatic environment, in the ocean really. So these are all our different shapes. Again, I'm apologizing that we're moving pretty quickly today. I have a little counter here in the corner. We're on slide 5 of 75, so we've got quite a ways to go. But if you do want me to repeat something, go ahead and stop me. Okay, so we're going to look here. Now here's the coccus shape there, and which are the little circles. Now, all of the different bacterial cells can also come as groups. So when they're pairs, they're diplococci or diplobacilli. And that di, that dip, or well, diplo, that di means two. So when you know that, you can kind of start to figure out what these words mean. Clusters are staph. So we have staphylococci. So when you are hearing about a disease and you say he has a staph infection, well, now automatically I hope your brain will go to, oh, that is a cluster of coccus-shaped. Chains are in streps. So streptococci, streptobacilli. So again, somebody says they have strep throat, you can say, oh, they have a chain of bacteria, because I know that's exactly where your brain will go. Groups of 4, so we can actually start to group these, and they almost start to look like grapes, actually. Will be tetrads. And then we can also have cube-like shapes where they're growing on top of each other. That's sarcinae. Yeah, so let's see. We can see all those different pictures. You've got your diplococci, your streptococci that's spread out, the tetrad, the sarcinae, and then the staphylococci, which really look like a group of grapes. This is just one extra slide on bacillus, where we have a scientific name that would be bacillus, and the shape is also bacillus. And this is a stain of, this is a figure from the new book showing that this is a nice chain structure of bacillus bacteria. It appears like they are staining gram positive because it's mostly that purple color. All right, so we're going to start to look at the structure of a prokaryotic cell. We'll look at all the different features. We're going to start on the outside of the cell and work our way in. We're going to start with, again, those structures that are external to the cell wall, outside of the cell wall. So outside of the cell wall, we find the glycolax. The glycolax, glyco, means sugar, so we know that this is going to contain some kind of carbohydrates. It's external to the cell wall. It is relatively viscous and gelatinous, so it's kind of slippery, and also used for attachment. It's made up of polysaccharide and or polypeptide. So it's got polysaccharide, poly means many, saccharide would be sugar. And then and or polypeptides, so it can be many proteins that are stuck together. And there's 2 different types of, oh, there's somebody coming in. Okay, there's 2 different types of the glycolax. We have the capsule, which is neatly organized, and it's very firmly attached. We can stain for a capsule. It's going to be just a very rigid, regular shape. Or we can have a slime layer, and I like the name, that's fun, slime layer, but it's unorganized and loose. It's going to have some lipids stuck in there as well. And it's not easy to stain because it's not going to be this nice regular circle around there. It's going to kind of stick out a little bit more. A slime layer is very useful for biofilm formation. So the glycolacs can contribute to virulence, where the capsules will prevent phagocytosis. Phagocytosis is the engulfment of a bacterial cell by an immune cell. And we do have 2 chapters on immunity, so we're going to be talking a lot more about phagocytosis coming up in the next, I think it's a couple of weeks from here. So there's some extracellular polymeric substances that will help to also form biofilms. And once we form a biofilm, the cells are protecting each other, and they're going to also aid in evading the immune system. So that's all I have on the glycolacs. Now let's move on to the flagella. So not every bacterial cell will have flagella, but some do. And what a flagella is, it's a filamentous appendage that's external to the cell. And it's there to propel the bacteria, to move it around. And it's made up of a protein that's called flagellin, which is why we call it a flagella. It's because the name of the protein. There are 3 parts to the flagella. There's an outermost region known as the filament. So the filament is the long part. Look at that. The hook is what is going to attach to the filament. So the hook is external to the cell wall. It attaches the filament to the basal body, which is that 3rd part, which consists of a rod and pairs of rings. And what it's going to do by having those rings is anchor the whole flagella into the cell membrane. So you can see in this picture here, you've got the filament, that's the long part, the hook external, and then you've got your anchor with 4 basal body rings. Bacteria have 4 basal body rings on their flagella. So it's really well anchored. And here is an animation for you to watch.
The long whip-like portion of the flagellum is called the filament. Filaments are composed of many identical globular protein molecules called flagellin. Molecules of flagellin form helical chains around a hollow core. Certain pathogenic bacteria can be identified and classified by differences in their flagellar proteins. In contrast with eukaryotic flagella, no membrane covers the filament of a prokaryotic flagellum. The whip-like filament inserts into a curved structure called a hook. The hook is composed of a different protein and connects the filament to the basal body. The basal body is composed of different proteins than the filament and hook. The basal body is composed of a rod and a series of either 2 or 4 rings, which anchor the flagellum to the cell wall and allow the flagellum to rotate 360 degrees. You're viewing a flagellum from a gram-negative cell. A gram-positive cell would only have 2 rings in its basal body, both of which are attached to the plasma membrane.
All right. So in addition to how the flagella is attached, we can have different arrangements of the flagella. It can be just one, it can be many, they can be on polar ends, or they can be all around. So we have in A, we have the petrichus, and that one is all around. Just the one tail, we have monotrichus. We have the lophotrichus and polar, where you have many of them and they're just at one end. And then we can have amphitrichus and polar, and that is where we have them at the polar ends, and there's many of them. So there's many different ways that we can have these flagella. Totally depends on the type of bacteria that you're looking at. That's another way to help classify bacteria. All right, so we're going to let this guy talk to you about those.
Cells with a single flagellum are monotrichous. When the flagellum rotates counterclockwise, the bacterium runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Amphitrichous bacteria have a flagellum at either end of the cell. Bacteria with a tuft of flagella at one end are called lavatrichous cells. When the flagella rotate counterclockwise, the flagella bunch up and the cell runs. When the flagella rotate clockwise, they separate and the cell tumbles. Paratrichous bacteria have flagella covering the surface of the cell. Runs occur when all of the flagella rotate counterclockwise and become bundled, while tumbles occur when the flagella rotate clockwise and separate.
All right, it's a quick animation, but it's very nice. So we are recording this, so you can go back and watch some of these. I have not been able to figure out how to get these videos out of this, so you can watch them separately. But I think it's very nice to be able to see how these bacteria move. All right, so in addition to how they move, why they're moving is going to be based on the type of stimulus. So flagella are going to allow bacteria to move toward or away from stimuli, and that stimuli is known as taxis. And so it can either be that it is a chemotaxis, a chemical that's maybe drawing it in, and immune cells will also use that. They'll use some kind of chemical taxis stimuli to bring bacterial cells towards them so they can be engulfed, so they can phagocytize them. Or it could be a type of nutrient, or it could be another cell. So maybe you have taxis of a bunch of biofilm cells. They're sending out taxis stimuli to say, come join our biofilm. So those flagella, they can run or tumble, as it said in the video. When you have counterclockwise, you've got all the flagella working together and the cell will run. Or it's going to tumble when they're clockwise and the flagella spread out. These flagella proteins are H antigens. So an H antigen is recognized by the immune system as an invader. And we can use this H antigen to distinguish among different types of bacteria or serovars. So for example, when we have E. coli, not all E. coli are pathogenic. And if they are pathogenic, there's going to be many different groups of them. So one of the ways to know the different types of pathogenic E. coli would be by the H antigen. And they are simply numbered. So the H stands for the H antigen. And then the number following is the category of H antigen that we have assigned it. So in this case, they're saying H7. So this happens to be the number 7 antigen in our category. There's H1, 2, 3, 4, 5, 6. I'm not going to count forever, but I think you get the idea. There are many different of these H antigens. By knowing the type, the very specific type of E. coli, we can better treat an infection. So here's some more motility.
Motility is the ability of an organism to move by itself. The motility of a cell may enable it to move toward a desired environment or to flee from a harmful environment. Bacterial cells use structures called flagella, or in the case of spirochetes, flagella-like structures called axial filaments to enable this movement.
All right. So if you were to be viewing under a microscope a bunch of live cells, maybe you'd see something fun like this.
Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus and more frequent when the cell moves away from the stimulus.
All right. So that's a lot of motility. We're going to look at archaea motility now. So archaeal cells have motility structures. They are made up of glycoprotein archaeolins. They're anchored to the cell. Archaeola, sorry, a little of a tongue twister there. Or singular, archeleum, rotate like flagella. Okay, axial filaments, they were mentioned briefly. These are also called endoflagella. Endo means within. So it's a flagella within the cell. And it's not actually inside of the cell. It is outside of the cell wall, but it surrounds the cell. And you can see that in this diagram here, those little ropes. And so it's contained with the cell. It's not going to be its own structure outside of the cell like we saw in those other pictures. They're found in spirochetes. They're anchored at one end of the cell. And as they rotate, they'll cause the cell to move like a corkscrew. So the flagella isn't going to detach and wave at any point. It's going to stay on the cell, but it's still going to allow movement for that cell. So here's an animation of that.
Spirochetes are a group of bacteria that are spiral-shaped. They move in a corkscrew-like fashion, using flagella that do not protrude from the cell wall like typical flagella. Lyme disease is caused by a spirochete called Borrelia burgdorferi. Another spirochete, Treponema pallidum, causes syphilis. Spirochetes have an axial filament that lies between the outer membrane and the cell membrane. The axial filament winds around the outer edge of the cell. The axial filament is made up of a bundle of modified flagella called endoflagella. Rotation of the endoflagella causes the axial filament to rotate around the bacterium, which causes the spirochete to corkscrew through its medium. It is thought that this manner of movement allows the spirochetes to move more easily through viscous human tissues and fluids, such as mucus. Then they can easily attach to the cells below.
All right, he's done. So those are, they're kind of creepy little guys. And like you said, there are pathogenic bacteria like this. And they, oh gosh, when you isolate them, they are goofy, scary looking. All right, so some other structures outside the cell wall include the fimbriae. And these are hair-like appendages, and they allow for attachment. These are particularly useful in attachment for to another cell, another bacterial cell for, again, biofilm formation, or to a host cell to infect it. Now, Pili can be involved in motility with a gliding or a twitching motility. But more often when we talk about pili, we're going to be talking about conjugation pili. And these are involved in DNA transfer from one cell to another. And so conjugation pili, it can reach out to another cell, and then DNA from the donor cell, it spirals out. Excuse me, I'm drinking a pop that's very bubbly. The DNA spirals out of the donor cell through the pili into the receptor cell, where the DNA can then be, the genes can be mixed and matched. This is a form of sexual DNA transfer between bacterial cells. Most of the time, cells, they're going to divide through binary fission, so they're going to be clones of one another. So this is a good way to get DNA transfer. All right, so those were all the structures outside of the cell wall. Now we're going to talk about the cell wall itself. So the cell wall is very important to prevent osmotic lysis and protect the cell membrane. So osmotic lysis means bursting. So lysis is breaking open, and osmotic is usually by water. The cell wall is made up of peptidoglycan in bacteria. There are two exceptional cases to that we're going to talk about. So they're unusual cell walls, but we'll get to it in a minute. And then the cell wall also helps to contribute to pathogenicity. So it's going to be able to help evade the immune system, or there's actually some toxins in the gram-negative cell wall. So composition and characteristics. Here we go with the peptidoglycan. Peptidoglycan is a polymer of a repeating disaccharide in rows. And there's this disaccharide is made up of NAGs and NAMs, N-acetylglucosamine and N-acetylmuramic acid. And so we have these two, and there's the structure, the chemical structures over there, linked together. There's an oxygen in the middle, and they can be hydrolyzed, so you can cleave that. But generally speaking, we're going to, if you don't have an antibiotic in there, these are pretty strong structures. They're tightly bound to each other, and then they are create these rows and rows that are linked by polypeptides. So they're going to be linked with some proteins. Gram-positive cell walls contain thick peptidoglycan cell walls. So there's a lot of peptidoglycan in there. They also contain some teichoic acids, which are going to aid in the structure. The teichoic acids include the lipoteichoic acid. That's going to link the cell wall to the plasma membrane, so it's going to anchor it. Wall teichoic acid will link the peptidoglycan to itself. These acids carry a negative charge, and they also aid in the regulation and movement of cations. So the cations that need to come through that cell wall, through the cell membrane for metabolic processes. Both the polysaccharides and the teichoic acids provide antigenic specificity. So the more, or the differences that you have in these chemical components will aid in how they evade the immune system. And so we've characterized them based on cell wall components. Gram-negative cell walls, on the other hand, now they do have peptidoglycan for sure, but it's a very thin layer. These gram-negative cell walls contain an outer membrane, and the outer membrane is made up of polysaccharides, lipoproteins, and phospholipids. So you see at least two of those words has a lipo in there, which means a fatty acid. So that's one of the reasons we can call gram-negative cell walls slippery. It's because they have a lot of lipids in there. And with those lipids, it helps to evade the immune system. They're very good at evading phagocytosis. There's a periplasmic space, and this periplasm is between the outer membrane and the plasma membrane containing the peptidoglycan. So there's an extra layer of protection almost, where if you're going to try to attack a gram-negative cell wall with an antibiotic, it not only needs to deal with a very slippery cell wall, it also has to deal with the periplasm before it can even get to the cell membrane to break that cell open and lyse it, kill it. Excuse me. So these gram-negative cell walls, all those components in that cell wall will help to protect from phagocytes. So phagocytosis, those cells that are trying to come around and eat the bad cells. Gram-negatives are very good at evading that. Gram-negative cell walls help to evade complement. Complement is a type of immune reaction that it's in the innate immune system, and we're going to get to that. We have a whole chapter on it. But basically what complement does is it activates a lot of different factors to break open unwanted cells. And also antibiotics. So gram-negative cell walls are very specific to the antibiotics that we have. We don't have many antibiotics against gram-negatives, so we do tend to hold those in reserve until we know for sure it's a gram-negative infection. So these cell walls are made up of the lipopolysaccharide LPS O polysaccharide. So the O polysaccharide functions as an antigen. And so, for example, we're looking back at our E. coli where we have the O polysaccharide as O157. Again, that number that comes after the O is just a category. So obviously there's at least 157 O polysaccharides that we have as humans, as scientists in our database. And so when we are looking at the particular E. coli, we say, oh, it's the O polysaccharide 157, and it's our H antigen number 7. So obviously these numbers can change. It depends on the type of E. coli that you're looking at. But the O and the H will stay the same. Lipid A, this is an endotoxin that we find embedded in the top layer. And this is one of the reasons we don't necessarily always want to break open gram-negative cells or lyse their cell walls, because it releases this toxin. And if you have a really bad gram-negative bacterial infection and you go and you treat it really aggressively, you're going to break all of these cells and you're going to have a whole lot of this toxin released, and that can lead to sepsis. So it's a smart idea to know if you have a gram-negative infection, first of all, and then secondly, to track fevers so that you know when the cells are dividing, and then possibly to treat it without a lot of antibiotic at first. So, ramp it up over time and really watch out for that sepsis. And also antitoxin drugs, you can give those. There are also some porins, which are made up of proteins that form channels through the membrane. And so these porins are very specific to allow nutrients or anything that needs to get in from metabolism of the cell. And they just surround a little hole, and they're very specific to what can get through. So the gram stain mechanism, we have done stains already, but just really in brief, we have the crystal violet iodine crystals. They form when you have the peptidoglycan. So in a gram-positive, the crystal violet cells are not going to leave. The decolorizing agent, alcohol, is just going to dehydrate that peptidoglycan and hold on to the crystal violet even tighter. Whereas the gram-negative, the decolorizer alcohol will dissolve the outer membrane, leave holes in the peptidoglycan, and the crystal violet washes out. And then safranin is your counterstain. So gram-positive cells will always appear purple, gram-negative as red if you did the stain correctly. So gram-positive cell walls can produce some exotoxins. So exo means out. So this is a big difference when you're treating gram-positive versus gram-negative. So like I said, with the gram-negative, the endotoxin, the toxin is held within that cell wall. It's not just out there attacking. So you don't want to break those cells all immediately and flood the system with that toxin. On the other hand, gram-positives, they're producing exotoxins. They're spreading their toxins out. So you actually do want to kill them faster. Gram-positive cell walls have a high susceptibility to penicillin or other antibiotics that are similar to penicillin because they target peptidoglycan cell walls to lyse the cells. These cell walls are also disrupted by lysozyme. And lysozyme is an enzyme that we all produce. It's found in tears. It's found in mucous membranes. Sweat has some lysozyme in it. So, you know, you actually, your hands are killing bacteria as you touch things, which is kind of neat. So again, these gram-negative cell walls, they have that endotoxin. Not all, but some also produce exotoxins. So they're doubly bad. And they have a low susceptibility to penicillin. Now, we do have antibiotics to treat gram-negatives, but when you're treating a gram-negative infection, it's going to be very different than a gram-positive. Okay, so here we are looking at the atypical cell walls. So as I kept mentioning earlier, we have a couple of outliers for this bacteria peptidoglycan blanket statement. And these are our acid-fast bacterial cell walls. They are kind of like gram-positive cell walls in that they have a lot of their particular chemical component stacked together. They don't have a lot of lipids, but they do have one particular lipid, and that is our waxy lipid mycolic acid. And this is bound to the peptidoglycan in such a way that we use a different stain for them. And these are the Mycobacterium and the Nocardia. And these are going to stain with carbofuran. So Mycobacterium, this is the picture that you're looking at here. They're going to be acid-fast positive, and it's for if you were looking for tuberculosis. Okay, here we have some other atypical cell walls. Now, these are not bacteria. Mycoplasmas lack cell walls altogether. They have sterols in the plasma membrane. So mycoplasmas are a group of unique microbes that are prokaryotes, but contain sterols like a eukaryote, and they don't have cell walls like animal cells. So they're just a group on their own. And we'll leave it at that. I don't think, there's just not a lot known really about them. The archaea can be wall-less, or they have walls of pseudomurin. So they aren't going to have the same components as a bacterial cell wall. They're going to have pseudomurin in there. They stain differently. And archaea that have cell walls, we typically find those in very extreme environments, like in Yellowstone Park in the really hot pools there, or in really cold areas. And those would be the walls of pseudomurin really protect the archaea. But some archaea don't have cell walls at all. So damage to the cell wall. Lysozyme can hydrolyze bonds in peptidoglycan. So if you have a really thick peptidoglycan layer, lysozyme's easily going to kill that cell. Penicillin will inhibit the peptide bridges. Again, it's going to break up peptidoglycan cell walls. A protoplast is a wall-less gram-positive cell. A spheroplast is a wall-less gram-negative cell. Both the protoplast and spheroplast are susceptible to osmotic lysis. So if a cell is in these stages and it doesn't have its cell wall on yet, it is susceptible. L forms are wall-less cells that swell into irregular shapes. So a cell that doesn't have its cell wall and is taking on water without regulation is called an L form. Okay. So now we're moving to the structures that are internal to the cell wall. So the plasma or cytoplasmic membrane. This is a phospholipid bilayer that encloses the cytoplasm. It contains some peripheral proteins on the membrane surface. Peripheral means on the edge, like peripheral vision, right? And so these peripheral proteins sort of sit on the top of the cell membrane, and they can be easily detached. There's also integral and transmembrane proteins. Now those integral integrated into, and transmembrane means trans is from one side to the other, like the transcontinental highway, or I think it was a railroad actually initially. So it goes from one end to the other. And so the transmembrane proteins go all the way across. And both of these types of proteins penetrate the membrane and have different functions. Integral proteins are usually for stability. Transmembrane proteins are usually for transport of metabolites or anything that's needed from one side to the other. So here we have an animation.
The cytoplasmic membrane of a prokaryotic cell is the main gateway for movement of molecules in and out of the cell. This membrane may also be called the cell membrane or plasma membrane. Cytoplasmic membranes in bacteria are comprised of a phospholipid bilayer. Each phospholipid contains a phosphate head located on the outer part of the bilayer, attached to a glycerol and 2-fatty acid, which face the core of the membrane. The phosphate is negatively charged and hydrophilic, while the tail portion of the phospholipids is hydrophobic. Thus, cytoplasmic membranes are very effective at blocking passage of many molecules, while other molecules are allowed to pass. Cytoplasmic membranes also contain 2 types of proteins. Integral proteins often span the entire bilayer and act as channels for transport of molecules that normally wouldn't be able to penetrate the bilayer. Peripheral proteins are loosely attached to the membrane on one side or the other. Some of these may be glycoproteins, which are proteins bound to a polysaccharide. These proteins may function as recognition sites, enzymes, or receptors.
All right. So the fluid mosaic model, I'm sure this was touched on in biology, but it's a very important model to discuss. This membrane, the plasma membrane, is as viscous as olive oil. So if you have the difference, if you're holding on to a thing of lard, or let's go with Crisco, because lard is gross. So you have Crisco, how thick that is, and you have your olive oil, which just can flow through your fingers. Now imagine, you just saw that video, how many things are moving in and out of the cell membrane, how some of those transmembrane integral proteins were, they had to change their shape in order to move something from one side to the other. In addition, just cell signaling itself, for the cell to send signals within the cell or out of the cell to tell it whether to stay or to go, or some different types of functions. All of these cell signaling, they need to be able to move in the cell membrane. They have to be free to move. And if you have something as thick as Crisco, you can imagine these proteins would be just stuck. They would not be able to go anywhere. They wouldn't be able to do anything. So that's why we have this nice olive oil phospholipid membrane. And that is due to having a double bond. So you don't have the saturated lipid. You have the unsaturated that have the double bond. So functions of the plasma membrane. The plasma membrane has selective permeability. This allows the passage of some molecules, but not others. So obviously we don't want everything just flowing in and out willy-nilly. You really want to make sure that you have specific metabolites coming in, specific markers going out, so on and so forth. The cell membrane contains enzymes for ATP production. Now this is in our prokaryotic cells. The enzymes for ATP production in eukaryotes is going to be in an organelle. I don't know if anybody wants to unmute and shout out that organelle. Nope. Okay. Well, that would be the mitochondria. So we'll be getting to that. But in a prokaryotic cell, there are no organelles. So the plasma membrane, the cell membrane itself, has to have these enzymes for ATP production. Some membranes have photosynthetic pigments on foldings called chromatophores. And those would be found in prokaryotes. Here we are. So we're going to get into membrane permeability. We have a number of slides. We'll let this guy start.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
Okay, so when you damage that plasma membrane, you can do this using alcohols. So that's very specifically 70% ethanol is how we clean our stations. You've seen a lot of hand sanitizer around right now, and that hand sanitizer should be 70%. If it's over 70%, it's not going to be effective, not as effective. If it's under 70%, it's not as effective either. You have to have that water in there. And that can damage the cell membrane. It's also going to damage the cell wall. Quaternary ammonium, also known as detergents, that can damage the cell wall. And then we have different types of antibiotics. Polymyxin antibiotics will cause the cell contents to leak out of the cell. So all of these things can damage the cell membrane. So as that video indicated, we were talking about the movement of materials, and we're going to talk about these now. There's two different processes following to concentration to a low concentration, and no energy is expended. So no ATP is used. In the active processes, substances move from a low concentration to a high concentration, and energy is expended. So we're going to start with the passive processes. The different types of passive processes. Simple diffusion is the movement of a solute from an area of high concentration to an area of low concentration. And this will continue until molecules reach an equilibrium. Oxygen is a very good example of simple diffusion.
Passive transport includes diffusion, nonspecific and specific facilitated diffusion, and osmosis. All of these processes occur along electrochemical gradients and do not require the expenditure of energy in the form of ATP. Diffusion is the net movement of a chemical down its concentration gradient, that is, from an area of high concentration to an area of lower concentration. This diffusion requires no energy expenditure by the cell, as the concentration gradient drives the diffusion. Small uncharged molecules, such as dissolved oxygen, dissolved carbon dioxide, small alcohols, and small fatty acids, can freely diffuse through the cytoplasmic membrane. Once equilibrium is reached, molecules will still move across the membrane, but there is no net movement in a particular direction.
Another form of passive process is the facilitated diffusion. In facilitated diffusion, the solute will combine with a transporter protein in the membrane. This transporter protein will transport ions and larger molecules across a membrane with the concentration gradient. Again, no energy is expended, no ATP is used.
Simple diffusion does not require special membrane proteins for movement of molecules. However, other types of diffusion do require such proteins for the transport of molecules. Non-specific facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of non-specific facilitated diffusion, these permeases allow a wide range of molecules that meet charge or size criteria to pass through their channel. Some facilitated diffusion is passive transport allowed by transmembrane proteins called permeases. In the case of a specific facilitated diffusion, these permeases only allow specific types of molecules to pass through their channel. The channel contains a recognition element. If the molecule is not recognized, the channel will not open to allow the molecule to pass into the cell. Osmosis is a special kind of passive diffusion that involves the movement of water molecules across a semi-permeable membrane. Water passes through the phospholipid bilayer with some difficulty, but transmembrane channels can ease the passage of water through the membrane in a process called facilitated diffusion. Water plays a critical role in maintaining cell integrity. Water tends to move across the membrane from areas of high concentration to areas of low concentration, just like solutes. The difference is that water makes up most of the liquid, so a change in the total concentration of solutes is really the force that causes water to move. If the concentration of solutes outside the cell is high compared to the concentration inside the cell, water may leave the cell, causing the cell to shrivel up. Similarly, if the concentration of solutes inside the cell is high relative to the outside, water may move into the cell and can cause it to swell up and possibly burst.
So as I mentioned, water is really following solute. And that's a good take-home message for everyone to remember. And that's why they say drink electrolytes. Don't just drink water all the time. If you're working out really hard, definitely have that bottle of water, but you need to make sure you have solutes, because the same thing will happen to our cells that we see are happening here to these microbes. The cells can burst or shrivel up depending on whether or not you have solutes there. So osmosis here is a passive process, and it specifically includes the movement of water across a selectively permeable membrane from an area of high water to an area of low water. However, that's going to be driven by the solute concentration. This is through the lipid layer. We have aquaporins, and aquaporins are water channels. So here we have the different solutions, and these solutions are talking about the solutes. So in an isotonic solution, we have the solute concentration is equal inside and outside of a cell, and water is at an equilibrium. In a hypotonic solution, solute concentration is lower outside than inside of the cell. Water moves into the cell. And in that case, we might end up with a cell bursting, right? In a hypertonic solution, solute concentration is higher outside of the cell than inside, and water will move out of the cell will shrink. Now, a hypertonic solution is what we often find microbes in if they are in a high salt environment. It's called a halophile, so it likes salt. And so these microbes they've developed a way, they've evolved a way to exist in these conditions in a hypertonic solution without the cell dehydrating. So let's move on to our active processes. Active processes are going to require energy, ATP. So they'll require a transporter protein, ATP, and can go against the gradient. So it can go from areas of a low concentration to high concentration. It can go uphill. Active processes also include group translocation, which requires a transporter protein and phosphoenolpyruvic acid, or PEP. And the substance is altered as it crosses the membrane. So if you need to grab something really big and move it from one side to the other, and you're going to change its shape, that's also going to require energy.
Active transport requires the cell to expend energy, usually in the form of ATP, to transport materials across the cytoplasmic membrane. This transport occurs through transport proteins and is usually against the electrochemical gradient, like carrying a load uphill. There are 3 types of transport protein. Uniport proteins transport only one type of molecule. Antiport proteins transport 2 types of molecules, but in opposite directions. Symport proteins transport 2 different molecules at the same time, but is coupled with a uniport channel.
All right, so we're done with transport. We're going to move on to the cytoplasm. So we're going to go inside of the cell now. The cytoplasm is the substance inside of the plasma membrane. 80% of the cytoplasm is water. It also contains proteins, carbohydrates, lipids, and ions. Inside of the cell membrane, sort of right underneath it, and then also, depending on the type of cell, we have the cytoskeleton. The cytoskeleton, those are very specific types of proteins that are involved in cell division, that are also involved in cell shape. Again, we are on prokaryotic cells, so we're going to talk about the nucleoid. So bacteria cells do not have a nucleus. They do not have a membrane-bound genome. So what we call this, the bacterial genome, it's not bound. It is free-floating in the cytoplasm, but we do call it the nucleoid. A bacterial chromosome is a circular thread of DNA that contains the cell's genetic information. Bacterial cells also include plasmids, and these are extra chromosomal genetic elements. These can carry non-crucial genes. I say can because I would argue that antibiotic resistance, or the production of toxins, or even some of these little plasmids will contain genes for survival in extreme environments. So I'd say they are actually kind of crucial, but they're easily shared. And so in this picture, they're showing a plasmid down there, and it's just a tiny little circle of DNA. And these plasmids are pretty easily shared from cell to cell. They can use that pili and send a plasmid to another cell. There's also viruses that infect bacteria can easily pick up plasmids and transmit it from cell to cell. So this is one way that antibiotic resistance can spread very quickly, is through this plasmid transfer. Ribosomes. So all prokaryotic cells contain ribosomes, and these are the sites of protein synthesis. So if you cannot do protein synthesis, the cell is not alive. Ribosomes are made up of protein, and of ribosomal RNA, or also known as rRNA. And these ribosomes are smaller than eukaryotic ribosomes. They are 70S, and that's a 50S plus 30S subunit that must come together before protein synthesis can occur. Now, I know everybody's still paying attention, and you're all saying, wait a minute, 50 plus 30, that's not 70. And you would be right. And that is because the S stands for sedimentation rate. So if you were to centrifuge the ribosome as a whole, it's going to sediment out at the number 70. If you have the separate subunits and you centrifuge them, they're going to settle out. They're going to, their sedimentation rate will be 50 and 30. So it's not actually adding them together on how big it gets. It's just the sedimentation rate of each component. All right, so many prokaryotes also have inclusions, inclusions. These inclusions are kind of vacuoles inside, and they can contain metachromatic granules, which is, oh yeah, no, I have magnetosomes in that picture there. And these contain phosphate reserves. Phosphates are very important to put that phosphate onto ATP, so you have to have a number of those reserves. Polysaccharide granules can contain energy reserves, so carbohydrates. Lipid inclusions would contain energy reserves of fatty acids. Sulfur granules would contain energy reserves of sulfur if your prokaryote used sulfur in its metabolism. Carboxysomes contain rubus, which is a coenzyme for carbon dioxide fixation during photosynthesis. When we get to, I believe this will be our next chapter on Wednesday, chapter 5, metabolism, we'll talk more about photosynthesis there. Inclusions are also gas vacuoles, and these can help the cell to maintain buoyancy in a liquid environment. And then finally, the magnetosomes. These contain iron oxide inclusions, and they can destroy water oxygen radicals such as hydrogen peroxide. And that's what we see in that picture. Endospores. Endospores are resting cells, and they are produced when nutrients are depleted. They are resistant to desiccation, heat, chemicals, and radiation. So, oh, I should have put on here, they're also resistant to the vacuum of space. Endospores are tough. We find endospores in a number of different cells, but for example, we're just using Bacillus and Clostridium here. Like Bacillus anthracis, you've heard of anthrax, right? Sporulation is endospore formation, and germination is when the endospore returns to a vegetative or metabolically active state. All right, moving on. We are checking the time here. We can do this. We're moving on to the eukaryotic cell. So this is a really fun picture of a eukaryotic cell where we have half plant, half animal cell. Those don't exist in nature. It's just a picture so that you can see all the different organelles. So eukaryotic cells contain membrane-bound organelles. They can have flagella and cilia. They can have cell walls, but those will have a different component than prokaryotic cells. So let's take a look. The flagella and cilia. Again, they are projections that are used for locomotion or moving substances along the cell's surface. Flagella are long projections, and they're usually few in number, where cilia are short projections, and they're quite numerous. So they look like little hair-like structures. So both flagella and cilia consist of microtubules, and these are made up of the protein tubulin. So this is a different protein than the flagellum protein that we found in the prokaryotes. So in eukaryotes, we have a different protein called tubulin. Microtubules are organized as 9 pairs in a ring plus 2 microtubules in the center. So we have a 9 plus 2 array. And these allow the flagella to move in a wave-like manner. So, cell wall. The cell wall in eukaryotes is going to be found in plants, algae, and fungi. It's made up of carbohydrates, where we have cellulose in plants, chitin in fungi, and glucan and mannan in yeasts. So I have the structures there at the bottom. We have a chitin structure and a cellulose structure. I didn't have room for the glucan and mannan. But you can see these are different structures. You have the nitrogens in chitin. Chitin is something that people tend to be allergic to. So you can have an allergic reaction to fungi. And we also find chitin in the exoskeleton of insects and shellfish. So chitin is an interesting allergen. So if you have a cell wall, it's going to have very different components. The glycolax of a eukaryote includes carbohydrates bonded to proteins and lipids in the plasma membrane, and it's found in animal cells. And we use this, it's sort of a new science, looking at the glycolax of animal cells, and it's a really interesting field with a lot of cell signaling involved. So the plasma membrane in eukaryotes is similar in structure to the prokaryotic cell membranes. We have the phospholipid bilayer. There's going to be integral and peripheral proteins. However, we have some differences in structures. All of our eukaryotes are going to contain sterols, which are a complex lipid. And they're also going to contain these carbohydrates for attachment and cell-to-cell recognition. And this is just a picture of a sterol of Leishmania, which is a multicellular animal parasite. Okay. So again, the plasma membrane is similar in function to prokaryotic cell membranes. We still contain that selective permeability. Simple diffusion, facilitated diffusion, osmosis, and active transport are all the same. So we're not going to rewatch all those videos or go back through that. We have the same function from prokaryotic to eukaryotic. Now the difference that we find in the plasma membrane here include endocytosis. So endocytosis, endo, and then which means within, and cytosis is bringing. And so this is phagocytosis and pinocytosis. So eukaryotic cells have the ability to grab something very large for phago or something smaller, and they will bring these things inside. So we have phagocytosis where they have pseudopods. So a eukaryotic cell, such as an amoeba, would have a pseudopod, which is a sort of foot that can extend and engulf particles. And it will do this, it's eating, that's what it's doing. In pinocytosis, instead of having the whole foot go out, something that might roll along the outside of the membrane can be folded inward. So it can bring in fluid and other dissolved substances. So that is a difference. Prokaryotes cannot do those functions. The cytoplasm is pretty similar. The cytoplasm is the substance inside the plasma cell membrane and outside of the nucleus. The cytosol is the fluid portion of the cytoplasm. The cytoskeleton is a little bit different because it has different types of proteins for eukaryotic cells. It's made up of microfilaments and intermediate filaments, and it's going to give shape and support, and it's also going to be very active in cell division. The cytoskeleton's pretty important in eukaryotic cells for that cell division because of all the organelles. Things have to be organized very specifically before a cell can divide. Cytoplasmic streaming is the movement of the cytoplasm through a cell. So ribosomes, again, the same as the bacterial cells, these are sites of protein synthesis. However, the ribosomes of eukaryotes are larger than the ribosomes of prokaryotes. They have a sedimentation rate of 80. So these are 80S ribosomes. Similar to prokaryotic ribosomes, they have two subunits, a large and a small subunit that must be bound together in order for protein synthesis to occur. The large subunit has a sedimentation rate of 60, and the small subunit has a sedimentation rate of 40. Ribosomes are membrane bound or free. When they're membrane bound, they'll be attached to an endoplasmic reticulum. When they're free, they will be free floating in the cytoplasm. Interestingly, eukaryotes also contain a prokaryotic type of ribosome, 70S, found in their chloroplasts and mitochondria. So this leads to the theory that prokaryotic cells engulfed each other to create the first eukaryotic cell a very long time ago. And so we have that theory coming up at the end. And so let's take a look at the organelles. The nucleus. The nucleus is the brain of the cell, a eukaryotic cell. The double membrane structure is known as the nuclear envelope, and it contains the cell's DNA. The DNA of a eukaryote is complexed with histone proteins to form chromatin. During mitosis, which is cell division for identical daughter cells, and meiosis, which is when we have the sexual genetic transfer of two different types of eukaryotic cells for non-identical daughter cells, chromatin will condense into chromosomes. The endoplasmic reticulum. The endoplasmic reticulum, it's a very folded structure. It has a transport network. There are two types of endoplasmic reticulum, the rough and the smooth. The rough ER is studded with ribosomes, so it's the site of protein synthesis. So we have the ribosomes synthesizing the protein, and then we have the ER folding those proteins directly. Many proteins are not functional until they are folded properly, meaning they have to have a specific three-dimensional shape in order to be functional instead of just a long chain. The smooth ER does not have ribosomes. It synthesizes cell membranes, fats, and hormones. The Golgi complex. The Golgi complex is important for the transport of proteins, so it is known as a transport organelle. It modifies proteins from the ER so that they can be transported via secretory vesicles to the plasma membrane. I think of the Golgi complex as the UPS store. And so it's got these proteins, it's packaging them perfectly and addressing them where they need to go. The mitochondria, the powerhouse of the cell. The mitochondria also contains a double membrane. The inner folds are cristae, and it has a fluid inside of it known as a matrix. The mitochondria is involved in cellular respiration, and ATP production. We are going to go into detail about this cellular respiration on Wednesday, so I won't do all of that here. We will talk a lot about the mitochondria on Wednesday. Chloroplasts. Chloroplasts are locations of photosynthesis. They contain flattened membranes that are thalloids, and these contain chlorophyll. If you'll remember, in prokaryotes, they just had their chlorophyll in the plasma membrane, in the cell membrane. In a eukaryote, there are very specific organelles that are dedicated to containing the chlorophyll and to ATP production led by light. Some other organelles that we find in eukaryotic organisms include the lysosomes. These are vesicles that are formed in the Golgi complex, and they contain digestive enzymes, so they are very useful in immune cells. The lysosomes will kill unwanted cells that have been phagocytized. So if it's going to kill the bacterial cell. Lysosomes are also useful to degrade proteins that are not folded properly. Vacuoles are cavities in the cell that are formed from the Golgi complex, and these are important to bring food into cells. They can also provide shape, and they can act for storage. The peroxisomes are very important. They oxidize fatty acids, and they destroy the oxygen-water radicals, including hydrogen peroxide. Centrosomes are networks of protein fibers and centrioles. They form the mitotic spindle, and they have a very important role in cell division. So they're going to line up the chromosomes so that you have that very specific cell division. So, our final couple slides, we made it. It's like we're 2 minutes away from 10:50, so we're going to make it. The evolution of eukaryotes. This is just the endosymbiotic theory is so cool. So life arose as simple organisms 3.5 to 4 billion years ago. And then the first eukaryotes evolved around 2.5 billion years ago. So the picture that you're looking at here are the cyanobacteria fossils that we find in Australia. Someday I'm going to get to Australia, and that's what I'm going to go and see. I mean, my family might go look at koalas, but that's what I want to see are those cyanobacteria. And these cyanobacteria, what they did is they were producing oxygen as a byproduct. Well, it got to a point where there were so many of these cyanobacteria that oxygen was slowly rising in the atmosphere that it created the Great Oxidation Event. And this oxygen ionized all of the iron in the oceans. The oceans weren't blue at that time. They actually were red. And so it created rust. And that rust layer, we can see it in the ocean. If you take a core sample of, and you can track it, you know where that was, and that's about 2.5 billion years ago during the Great Oxidation Event. So a lot of oxygen was released from the oxidation of this iron, and all of a sudden all of these organisms were saying, well, what do I do with this oxygen? And there were a few little guys that could use the oxygen to make energy. And so one engulfed another. You get your first eukaryote. So this is our endosymbiotic theory, where larger bacterial cells engulfed smaller bacterial cells, developing the first eukaryotes. And this happened twice. We have ingested photosynthetic bacteria to become chloroplasts, and then ingested aerobic bacteria to become the mitochondria. So this is my last slide. It is 10:50, and I need to get going because I'm going to go sampling on the lake with my students for research, and I hear somebody knocking at the door right now. So, whoops, I'm going to stop sharing and say goodbye to you guys.
Thank you.
Thank you so much.